Have you ever wondered why siblings do not look exactly alike given the fact that they have the same parents? To answer that, let us dig deeper and determine first how reproductive cells are made. This process is called meiosis. Meiosis is a cell division that creates gametes or reproductive cells. It comes in two consecutive cycles, namely meiosis I and meiosis II. It is also known as reduction division because the main goal is to reduce the number of chromosomes in half so that a haploid egg and a haploid sperm will be produced. Haploid cells contain 23 chromosomes. When the sperm and egg unite, a diploid organism will be produced. Now, remember that humans should only have 46 chromosomes, otherwise disorders like Down syndrome or trisomy 21 might occur. Maintaining the exact chromosome number is one tough job meiosis needs to accomplish. That's why preliminaries like the interphase, which ensures cell growth and DNA replication, take place before meiosis I, or the first cell division process of the whole meiosis cycle. After meiosis I, two haploid cells will be produced, and these will undergo interkinesis or cell rest before proceeding to meiosis II. By now, you might be wondering why I use chromatid instead of chromosome. So, before we go to the first stage of meiosis, let's first look into chromosomes, chromatids, and the centromere. Our genetic material is condensed into thread-like structures called chromosomes, or the X shape we see in cell illustrations. This structure, which has two arms and a centromere at the center, is called a chromatid. However, the presence of the centromere makes it an individual chromosome. During interphase, these are replicated, forming full-fledged chromosomes in X shape. Full-fledged chromosomes contain two chromatids, which are also called sister chromatids. Prior meiosis I, the cell undergoes replication, so we start meiosis I with 46 chromosomes. These 46 chromosomes pair up, forming 23 homologous chromosomes, which carry the same genes, each from your parents. Prophase I has 5 stages, and the first one is the leptothene stage, where the chromatins condense and chromosomes appear. In here, there are 46 chromosomes, but you will see only 4 to represent them. In the zygote stage, synapsis, or the pairing of homologous chromosomes, starts through the presence of the synaptonemal protein that connects the two. In the Pachytene stage, crossing over, or the exchange of genetic material in the chromosome segment of non-sister chromatids take place. Due to the crossing over, there will be genetic variation which make each of us unique or different from one another, even coming from the same parents. This is the reason why no siblings look exactly alike, except of course for identical twins. In the diplothene stage, the homologous chromosomes start to separate in the process called terminalization. However, strands of DNA are still connected at the site of exchange, forming an X-shaped structure called chiasma. The fifth stage is diakinesis stage, where homologous chromosomes continue to separate, and the chiasma undergo terminalization. Centrioles move to the opposite poles and form spindle fibers. Nucleus and nuclear membrane disappears, and the spindle fibers will attach to the kinetochore of the homologous chromosomes, and this marks the end of the prophase stage. In metaphase 1, pairs of homologous chromosomes move to the equator of the cell. Take note that we are still talking about 23 pairs of chromosomes, but you can only see 2 pairs to represent them. The imaginary line to which they assemble is called the metaphase plate or the equatorial plate. In anaphase 1, the homologous chromosomes separate and move to the opposite poles of the cell because of the shortening of the spindle fibers. The cell begins to elongate and form a cleavage furrow, an indication that the cell is now starting to divide. Take note that each side of the cell now has 23 chromosomes. In telophase 1, spindle fibers disintegrate, homologous chromosomes disperse, and the nuclear membrane reappears. Cytokinesis, or the separation of the cytoplasm, will happen next. At the end of meiosis I, 2 haploid daughter cells are formed. This means that each cell has 23 chromosomes, half of the original chromosome number of the parent cell. After cytokinesis, interkinesis will follow, and the cell takes its time to rest before proceeding to meiosis II.
Good day, everyone. This is a continuation in your previous discussion on Meiosis I. I will be discussing about Meiosis II, and I am your lecturer, Mr. Nestor Jr. C. Malilin. You've noticed that after Meiosis I, in the telophase 1, there are 2 produced daughter cells. But before proceeding to Meiosis II, there is a brief resting period between telophase 1 and prophase 2. This is called Intrakinesis. There is no duplication of chromosomes happens in interkinesis. Take note that the following events in meiosis II will be similar to mitosis, except that the daughter cells produced will have only half of their parents' genetic material. Let's start with the first stage in meiosis II. We have the prophase II. Notice that there are already 2 daughter cells that will divide in this stage. In prophase II, here are the significant events. In this process, the chromosomes begin to condense again. The nuclear membrane and the nucleolus starts to disappear. Lastly, the spindle fibers also begins to form. These are the important events happening on prophase II. After prophase II, we have metaphase II. The chromosomes will now start to align themselves in the equatorial plate. The kinetochore are then attached to the centromere of each sister chromatid. These sister chromatids prepare themselves to move on the opposite poles. Just remember that in metaphase, the chromosomes align themselves in the middle. The third stage is the anaphase II. In anaphase II, the sister chromatids begin to separate. Each sister chromatid moves to the opposite poles, which is evident in this illustration. Anaphase 1 or 2 may not correctly separate the homologous chromosomes and their sister chromatids respectively. The failure to separate the homologous chromosome is called non-disjunction. Non-disjunction may have an adverse effect depending on the chromosomes affected. In humans, when chromosome 21 experiences non-disjunction, the resulting consequence is a person with Down syndrome. People with Down syndrome have 3 copies of chromosomes 21. It means that there is an extra chromosome 21 instead of just 2. The 3 copies occur when one sex cell, either sperm or egg, with 2 copies of chromosome 21, because of non-disjunction. This is just an example when anaphase 2 or in anaphase 2, the homologous chromosome fails to separate properly. Lastly, we have telophase 2. In telophase 2, the chromosome starts to uncoil, and the nuclear membrane and the nucleolus reappear. At this point, cytokinesis finally splits the cell, producing 4 haploid cells. Thus, keep in mind that the products of meiosis are 4 haploid daughter cells, whereas the products of mitosis are 2 diploid daughter cells. This is the outcome after meiosis II. It produces 4 daughter cells which are not genetically identical, and the chromosome is half of the parent cell. So remember that meiosis is for sexual reproduction. Take note also that we have gametogenesis. In gametogenesis, it is a process of producing gametes or sex cells. There are 2 types depending on the sex cells involved. We have spermatogenesis. It is the process of producing sperm cells in males, whereas oogenesis is the process of producing egg cells in females. Spermatogenesis and oogenesis both involve meiosis. Let's start first discussing spermatogenesis. Look closely on the illustration. In spermatogenesis, the spermatogonium forms into the primary spermatocyte. The primary spermatocyte enters meiosis I to produce 2 secondary spermatocytes. The secondary spermatocyte enter meiosis II to produce 4 haploid spermatids. Spermatids undergo spermiogenesis, wherein spermatids mature into motile spermatozoa, which are the sperm cells. Thus, each cycle of spermatogenesis produces 4 sperm cells from 1 spermatogonium, which is evident in meiosis. Next is oogenesis. In oogenesis, it starts with oogonium. The gonium becomes the primary oocyte. The primary oocyte will enter meiosis I. The result of the primary oocyte is a secondary oocyte and a polar body. The secondary oocyte will enter meiosis II to produce 1 egg cell and another polar body. The first polar body produced in meiosis I divides to form 2 polar bodies and cannot be fertilized by a sperm. Thus, each cycle of oogenesis produces 1 egg cell and 3 polar bodies, all from 1 oogonium. The haploid sperm cell and the haploid egg cell produced in meiosis will meet during fertilization. The resulting fertilized egg is diploid, with half of the chromosome coming from each parent, 23 from the father or from the sperm, and 23 from the mother or the egg. Thus, in fertilization, the original diploid number of chromosome in a species is restored. So 23 from the father plus 23 from the mother, we have 46 chromosomes. And now, we will discuss about the cell cycle errors. Genetic mutations may happen in the parts of the DNA responsible for regulating the cell cycle. Mutations are changes in the nucleotide sequence of DNA which may occur in somatic cells or in gametes and be passed to the offspring. Mutation happens when DNA fails to copy accurately. Influences like exposure to specific chemical or radiation can also cause mutation. These agents cause the DNA to break down. We have chromosomal mutation. When we talk about chromosomal mutation, These mutations involve changing the structure of a chromosome or the loss or gain of part of a chromosome. The first example of chromosomal mutation is deletion. Deletion is due to a breakage or a piece of chromosome is lost. It is clearly shown in the image that a part or a segment of the chromosome is then deleted or removed, and then the genetic material is also lessened. Deletion can also have different effects. Example of an effect of deletion or disorder that results from deletion is Credoshat. Credoshat is caused by a deletion of chromosome 5p or chromosome number 5, which is also called as the 5p minus babies. Individuals with Credoshat have high-pitched cry, poor muscle tone, a small head size, and low birth weight. They also have problems with language and may express themselves by using a small number of words or sign language. The second example of chromosomal mutation is inversion. In inversion, chromosome segment breaks off, flips around, and then reattaches. As you can see in the illustration, there is an inversion that is happening on the segment of the chromosome, and then it is reinserted in the piece of the DNA. An example of disorder involving inversion is hemophilia. Hemophilia is a rare disorder in which your blood doesn't clot normally because it lacks sufficient blood clotting proteins. If you have hemophilia, you may bleed for a longer time after an injury than you would if you blood clotted normally. Next, chromosomal mutation is translocation. In translocation, it involves two chromosomes that are not homologous. Part of one chromosome is transferred to another chromosome. It is really evident in this image that the part of the white chromosome is transferred to the part of the black chromosome, or there is an exchange that is happening on the chromosomes. Example of disorders involving translocation is mental retardation, infertility, and cancer. The fourth chromosomal mutation is duplication. From the word self, there is another copy. So in duplication, it occurs when a gene is repeated or a section of DNA is duplicated. Lastly, we have non-disjunction. Non-disjunction is a failure of chromosome to separate during meiosis. We've discussed it earlier on anaphase. It causes gametes to have too many or too few chromosomes. So as you can see on number 3, on anaphase, when chromosomes fail to separate correctly, it can lead to too many or too few chromosomes from each of the daughter cells. Example is Down syndrome, also known as trisomy 21. It is also a genetic disorder that is caused by the presence of all part of a third copy of chromosome number 21. Up next is gene mutation. In chromosomal mutation, the affected is the chromosome, but in gene mutation, the affected is the sequencing of the nucleotide. We have first point mutation. Point mutation includes the substitution of one nucleotide in a gene. Example is sickle cell. Sickle cell is a result of one nucleotide substitution in the hemoglobin gene, which affects into the abnormality of the shape of the red blood cell. As you can see, on the right part, that is a normal red blood cell. But on the left part, it is an abnormal red blood cell. So when the red blood cell flows into the veins, the sickle cell can result to the abnormality of the blood flow or can block the blood flow, which will have an adverse effect to the human. Lastly, we have the frame shift mutation. Frame shift mutation is the insertion or deletion of one or more nucleotides. So there is a change in the reading frame, like changing a sentence. So proteins are built incorrectly. We have the first example. We have the ATG or the methionine, GAA the glutamine, GCA the alanine, CGT the glycine. If we remove one of the nucleotide, which is G, there will already a change. You can see below that the nucleotide will move to the left. So from GAA, it becomes now AAG. From glutamine, it will now turn into lysine. From alanine, it will now turn into histidine. So there is a change in the reading frame if one of the nucleotide is removed or inserted. So errors during cell cycle may occur beyond our control. Some of these bring harmful effect like disorders presented. But there are also mutations that cause no problem at all and in fact are beneficial. To end our discussion, take note that life is a cycle. There will always be a beginning and an ending, and it is continuous. Thank you for listening.